Hello, my name is Emre Seli. I am the Scientific Director of IVIRMA United States. I'm also a professor at Yale University. I want to talk to you about how we see mitochondria and the role of mitochondria within the context of reproduction as a potential target for diagnostics and therapeutics. Mitochondria are very important organelles for the cell, as they are the source of energy as well as many other functions. Within the cell, they are needed to be in stable condition. If they are not, and if they cannot deal with the stress that they endure, they will die, and then the cell will die or be unhealthy. Within the context of reproduction, a number of mitochondrial dysfunctions have been associated with decreased fertility, diminished ovarian reserve in animal models. These include mutations in mitochondrial DNA, problems with mitochondrial fusion and fission, which is division of mitochondria or fusion of mitochondria, and mitochondrial stress response. So from all these data, we know that mitochondria matter, and they matter for reproduction. Therefore, a lot of people concluded that they could be a target for diagnostic or therapeutic approaches within the context of assisted reproduction. As far as testing goes, a number of studies decide to investigate the copy number of mitochondrial DNA as a predictor of embryo transfer outcome in women undergoing IVF. Initial studies, some of them coming from really good laboratories, were really encouraging. However, further studies done in very stable laboratory conditions show that in most contexts, this approach does not really provide a lot of benefit. So we do not use it anymore in our clinical setting. Additional, more encouraging but still experimental testing approach is FLIM, which is the fluorescent lifetime imaging microscopy, which includes targeting the mitochondria with a laser, giving energy so they can fluoresce based on their energy content of NADH and FAD. Initial studies in our laboratory in collaboration with Harvard group has resulted in very encouraging findings, but they have to be validated in the clinical setting. As far as therapeutic approaches, as most of you know, a group in Harvard and a company that stemmed from that group investigated replacement of mitochondria from so-called ovarian stem cells into aging oocytes to improve oocyte outcome. Unfortunately, this didn't really pan out, and actually a randomized clinical study that was led by Elena Labarta at EV Valencia showed that this approach should not be used in clinical setting. Additional testing was done in collaboration with Marga Espert from EV Barcelona to see whether we could use rapamycin treatment in aging oocytes. However, the initial in vitro randomized clinical study did not show a lot of benefits. So we're still excited about the potential of mitochondria as a therapeutic or diagnostic target. However, I think we still have some time to achieve something that is clinically relevant. Thank you.